Thanks, Howard. Yeah, it's a pleasure to be here. I really want to thank the organizers, not for the opportunity to speak, but the fact that the humidity was lower than 50% last night when I went running. And the reason I say that is because I'm from Houston, Texas, and we're just about at the end of 3 months of summer, which is quite brutal down there. So just before I get started, the question, I guess, that this whole workshop is based upon is can the microbiome help health? And actually, I would argue that answer is already known. The answer is clearly yes. Certainly what I'm going to talk about today is the ability of the microbiota to impact Clostridium difficile disease. And unless you've been living under a rock, you probably know that fecal microbiota transplantation is a successful way to treat recurrent Clostridium difficile disease. So the answer, I would say, is definitely yes, but that's very low-hanging fruit. And I think the real question we have is, are we going to be able to manipulate long-term some of the more chronic and difficult to treat diseases? So as I mentioned, I'm at Baylor College of Medicine at the Center for Metagenomics and Microbiome Research. My laboratory is focused mainly on therapeutic microbiology, and we work in a number of different areas. We work with traditional probiotics. We also work on isolating next-generation probiotics, which is what I'm going to talk about today. But we also do a lot of work in genetic engineering of lactobacilli and expressing human proteins. We've just recently got a pretty good platform together for making and secreting high levels of human proteins, in some cases active. So if you have a favorite human protein you think you might need to make, would be happy to talk about it. So the big question that we're really interested in answering in the lab is really how do microbial communities resist invasion by pathogens? And as I mentioned, we'll talk about Clostridium difficile today. But because of the workshop nature of this talk, I wanted to spend some time talking about some of the challenges that we face in trying to do our basic science, but also do it in the context of whatever we find in the basic science realm we'll be able to hopefully translate. And I will say that the work that I'm going to talk about today was supported by this U19 grant. And I have to say that this funding by NIH was great because the reactors I'm going to talk about today were really just in theory when we applied for the money, and it's virtually impossible to get grants when you don't have 99% of your grant done before you actually submit it. So this work was done by 3 people here. So Kathy Robinson was a former graduate student in the lab who's moved on to the University of Oregon. Jennifer Ochtung was a postdoc in the lab, and she's still in the lab, but she now is assistant professor at Baylor. And then James Collins, who's a postdoc. And so all the work I'll talk about today was due to them. And so what we've really been trying to do in the last 5 years is trying to come up with new model systems for studying microbiome interactions with the host and also with pathogens. So today I'm mainly going to talk about these mini bioreactors that we've made, and that they're pictured on the left. We've also made humanized microbiota mice. So this is something that you're seeing increasingly in the literature, and these are just simply germ-free mice that have been colonized with human fecal material. And so we've been working with these for about 3 years, and they've actually been a pretty robust model for looking at complex host microbiota pathogen interactions. So Clostridium difficile is a big problem in hospitals now. It actually, in many hospitals, is becoming the most common cause of hospital-acquired infection, surpassing MRSA. It causes a nasty diarrhea, so if anybody's ever known anybody that's had this, and I have, it's not your garden variety food poisoning. I mean, this is very serious and often requires hospitalization. And it can progress on to death. And so one of the biggest problems with C. diff is that 15 to 25% of patients that actually get C. diff will end up having recurrent episodes, meaning that they can be treated with antibiotics, but within the cessation of those antibiotics, usually within a period of a few weeks, they will get recurrent disease. And some of these patients have this recurrent disease for years. And so you can just imagine how debilitating that is. Basically, they have no quality of life. As I mentioned, fecal microbiota transplantation has really been now the therapy of choice for these people. It's still not approved yet, but people are doing it because really they have no other option. And it is a big burden to the U.S. healthcare system. And so the CDC put together a really, I think, interesting report back in 2013 highlighting the emerging pathogens that are going to be a problem, and they just really highlighted here that C. diff is a big issue for infections that are occurring. So our primary goal is to try to figure out how to define communities that could suppress C. difficile infection so we could maybe supplant the need to do fecal transplantation, because there's clearly feces is undefined, and there's a lot of other things in feces other than bacteria. I know we talk about microbiome a lot here, and I think a lot of people use the term microbiome to talk about the bacteria that are in feces, but we've already heard about helminths today. We should be thinking a lot more about not only bacterial viruses, but eukaryotic viruses that are part of that. There's a lot of very interesting research showing how the innate immune system is educated by eukaryotic viruses, not just the bugs. So in that vein, we really wanted to try to create a model system here where we could actually look at sort of bacterial communities that can suppress C. diff. And I just show you here a sort of a typical model of a gut model that's used by a lot of laboratories, and a lot of the gut models that you'll see out there are people trying to build a gut. So you'll see here they have basically a 3-vessel model where they have, this is supposed to be your ascending colon, the transverse, and the descending colon. They have different pHs in the different stages. You can see that they have automated pH controllers, very big vessels here. So it's very complicated, and you can imagine that if you were going to use this system to screen, when they do their experiments, and I should mention that this system has been used for C. difficile experiments, they really only run 1 or 2 experiments per paper. It can take 60 to 90 days to complete a single experiment in this system for reasons I won't go into. But if you were going to try to screen dozens of therapeutics for this, you could just see that it's just not only cost ineffective, but it's also time ineffective for trying to do that. So really what we were interested in doing is trying to build a simpler model of C. difficile infection in vitro. And that's where we came up with the idea of mini bioreactor arrays. And so what we've built are these, essentially these plastic strips that have 6 individual chambers born out of them. These are actually built for us by a company, and essentially this gives you the dimensions. They hold about 25 mils. We usually run them at about 15 mils, so again, we're using a lot less media. Media tends to be expensive when you're running these continuous flow reactors. These reactors were built to fit on this 60-position stir plate, so if you have $3,000 burning a hole in your pocket, you can buy 60 magnets and they'll spin your cells for you. I didn't even know they made these things before we did this project. I had no idea why they made them before that. But so in any event, this is what they look like in practice. And so when they come to us, they're completely sealed. We drill holes in them, and we have an influent and an effluent port, and then we have a sample port. So essentially, we are cultivating these communities in these little reactors. And so we, again, they're not that expensive. For about $1,000, we can get 24 of these reactors, and they can be autoclaved and used multiple times before you have to get new ones. So the way we run these experiments in the reactors, you can think of them almost as a chemostat type of thing. We actually have simply a fresh medium here that's being pumped in and then pumped out into a waste bottle. The beauty of this is we can do this directly right in the anaerobic chambers. The chambers are preheated to 37 degrees all the time. We've come up with a way to modulate the pH so we don't have to worry about pH crashing in the experiments with buffer. And as I mentioned, they're continuously stirred, and they have about an 8-hour retention time. So I'm going to tell you about what you can do with these for C. diff and show you that there is some functionality of the human microbiome that we can reproduce in these small vessels, but certainly you could think about doing this for a lot of different applications. So this gives you an idea of the experimental design that we do. Again, mentioning that we have about 60 to 90 days to do it in a more traditional gut model system. What we do is we take human feces, we put them into the reactors. Because they're coming from frozen, we give them about a day to adapt to that environment, and then we basically turn on the pumps. We give them another day to sort of adapt to this flow and this continuous influx and efflux of sample. And then we either treat with clindamycin, an antibiotic that was traditionally associated with C. diff infection, or we mock treat as a control. We do that for 3 days. We give them a day off to let the antibiotic reduce down to low levels, and then we add Clostridium difficile. And then we just simply monitor C. difficile abundance and basically just sort of comparing what happens between the 2 different systems. So again, I think the beauty of this is that we can really do replication. So you can see here's an example. So when we put 10 to the 5th or so vegetative cells or spores into this, you actually see that if we mock treat, we actually see a washout of the C. diff. So this community now is resistant for invasion by Clostridium difficile. What I will mention is some experiments and some reactors, we see this go out by the rate of dilution, but we have other communities that seem to be actively killing off the C. diff, and they go away very quickly. So we're interested in that. You can see if we clindamycin treat these reactors, you can see that we get a nice steady invasion of the community. And if we test here, you actually can see spores being produced. You also see toxin being produced. So the C. diff is doing the same things that we don't want them to do in our colons as they're doing in the reactors. Now, hopefully the hospitals that people are staying at don't have 10 to the 5th CFUs of C. diff being put in their patients, so we were more interested in asking, could we go lower? And so we've actually went down to as low as 150 cells, and you can see that they'll invade, and they'll actually find their niche in this environment. And so that niche is about 10 to the 6 CFU per mil. Bioreactor media tends to be a poor medium because we don't, your colon hopefully is not getting high levels of glucose or simple sugars there. So C. diff only grows up to about 10 to the 7th in this medium. So it's doing actually pretty well in the presence of this complex community. So one of the things we get asked a lot, though, is so what do you do when you actually treat with clindamycin? Of course, we were worried about that too. Maybe we're just blowing up the community so much that there's just not bacteria there to compete with them. And this just shows you that over time, the amount of bacteria in either the treated with clindamycin or untreated, you can see that they're basically identical. So the bacteria, the community that's actually treated with clindamycin, because it's in this continuous flow, you don't actually lose biomass. You just get a different community that comes up, and that community can't resist C. diff anymore. That's really kind of the key point. That's a lot about what we think happens in people. What we do see, though, is we do see a drop in diversity. So you can see they start off at about the same diversity. So you can see here's our fecal samples. And so we get about half the diversity that we see in a fecal sample, which we think actually was quite good. There's a lot of things that are in feces that are probably not alive. So this is actually a very robust and diverse community. But you can see that the diversity has changed. So we actually have really changed the diversity by about half. So we have less than half the diversity, even though we have the same amount of biomass in our reactors. So that's probably speaking to where things are going. So you've seen a number of different plots today on how to look at these different communities. This is just an NMDS plot, and without going into the details of it, I think this is basically just clustering every one of these dots as a different bioreactor, and you can see that the clindamycin treated migrate away from here. And so essentially what we're interested in is who are the bacteria that are here that are now absent here, because these are going to be the representatives that are going to be most likely the bugs that we want to get out and try to cultivate as a defined community to suppress C. difficile invasion. So a number of these that are highlighted in bold have been associated with animal models of C. diff and also in human patients, and so we're pretty excited about these. And this basically is C. diff, and you can actually see some other organisms that are associated with disease, and Chronobacter has also been one that in animal models has been shown to bloom when C. diff infection is going. So it gives us a lot of the confidence that what we're seeing is in fact something that, is something, a normal function of a complex microbiota that would happen in an animal. So I want to tell you a little bit about how we've applied this to look at pathogenesis of C. difficile and learning about how epidemic strains of C. diff have come about. So the question that we asked is, are ribotype 027 strains of C. difficile more ecologically fit than traditional strains? So for those of you who aren't familiar with that, so in the last 15 years, ribotype 027 strains of C. difficile have swept across the globe. They started, there was an outbreak that was first documented in Canada in 2003, and these strains basically can be found in any developed country in the world now. They're associated with increased morbidity and mortality, and they're also frequently characterized as hypervirulent. So people have been studying the toxins Some people say they make more toxins. Some people say the toxins that are made are more virulent. But what more epidemiological studies recently have shown is that the disease severity does not normally cluster with ribotype. So what's not in question is the fact that this strain has made it all across the globe. And so we wanted to just simply ask, maybe these guys are just better at growing inside of a gut compared to more traditional strains, and that's why we see them so much more. So we basically used our same experimental setup, but rather than infecting with one strain of C. diff, we now are doing a competition between ribotype O27 strains and non-O27 strains. And so this just gives you an idea of one of the competitions we've done. So we have 5 sets of reactors here. This basically is the ratio of the O27 strain to the non-O27 strain. We started some at a 1-to-1 competition. We actually even put the O27s at a disadvantage at a couple here. And so if the O27 strain wins out in the competition, you'll have a ratio of higher than 1, vice versa if they lose, and you can clearly see that not only do these guys win, but they win quite big in a fairly short amount of time. So clearly the O27 in this competition won, but so the question would be, well, maybe you just got lucky and you picked 2 strains and this is how they worked out, so we went to additional clinical samples. And here I'm just showing you a day 7 time point, but again, the O27 strain here, and then there are different ribotypes of C. difficile that are not associated with epidemics or hypervirulence, and you can see in each case the O27 strains win. So we believe that the O27 strains have evolved in some way to be able to better compete in the microbiota, and that's something that we're actively investigating now. But this is a bioreactor, and there's no host component here. Everybody always points that out to me. I'm aware that there's no host component there. So that's why we then built these humanized microbiota mice. And so the motivation was, so there is a standard mouse model of C. difficile. Works great. The problem is that the bugs that are there are mouse derived, right? And so they've evolved for a long time. And so if you're going to ask human strains to go in there and compete with those guys, they're going to, ecologically speaking, they should lose, and they will lose. So what we've done is we wanted to use human microbiota mice. So we actually put human microbes in there, and then we're going to compete potential therapeutic communities with the human microbiota. And so we've done this, and I won't go into it, but certainly happy to answer during the break if anybody's interested. We bred these animals for 3 years. They're not kept in isolators. They're actually just in SPF conditions. What we found is once you put a complex microbiota in there, it's pretty resistant from getting invaded by other mouse commensals that are probably floating around the facility. They're a little bit less diversity than conventional mice, and they're definitely different community structures. If they weren't, we wouldn't be able to tell them apart. So it's a good thing that we did. But so what we've used these mice for, we've made a recurrent model of disease that we just published in Microbiome if you're interested in looking at the details there. But we wanted to ask, would this competition recapitulate inside these mice? And so it's essentially the same experiment. We use slightly different antibiotics. There's a 5-antibiotic cocktail followed by a single clindamycin injection that then C. diff is then added, and then we monitor C. difficile abundance. And so essentially we're looking at the ratio of the 2 different strains over time here. There's lots of details here that are tricky. You have to run the models in different ways. Sometimes when you add C. diff, you kill them. We obviously can't kill our mice because we want to see how the dynamics of the bugs go. So we use a slightly shorter course of antibiotics than normal. And so just to remind you, we did 2 of these competitions, and we were able to do these 2 because there are different drug resistances that are just normally found in these O27s versus these 2 ribotypes. And again, in the reactors, these 2 O27 strains really dominate over the other ones. And what was really gratifying is that in the mice, we basically saw the same type of dynamics between the strains. So again, giving us, good, you always worry that your model's contrived and it's not really going to recapitulate what's going on in vivo. And I think for us, this was a very gratifying result that what we're seeing is probably okay. So really what we're trying to do now, as I mentioned, we're trying to go ahead and get these defined communities. Now, there's companies out there that are already doing this. You can even see they're to the point where they're even sort of advertising on their website that they're doing this, which means they're pretty far along. But we've established a recurrent model of disease, and I think what really we want to do is try to figure out not only what communities are going to be required to suppress C. difficile, but can we also say these communities aren't going to cause something bad to happen, which I'll get to in a minute. And we also have a pretty good idea of what the molecular basis for this O27 strain is. It's actually the ability to eat a sugar, but I don't really have time for this conference to go into it, but we're getting there for that. So in the last few minutes, what I wanted to do is sort of talk about some of the problems that we think about a lot in trying to study in this field. Some of these, I think, are things that are now being more widely accepted. Some of them are some of my own personal pet peeves, so you have to put up with them. But I think that, with everybody jumping into the field, it's important to make sure that we're all talking about the same thing. And so one of the biggest things, if you don't remember anything from this talk, the functions of bacteria are strain and they're not species specific, okay? So if you see a Lactobacillus reuteri in your gut microbiome, or you see something that sounds like a pathogen, that doesn't mean that's the case, and I'll get to that in a minute. So many of the bacteria we're identifying have not been cultured before, and that's kind of where we're stuck in our project right now. So we know what bugs we want to get out, but we're having to struggle to get some of them to grow in pure culture. And so this is what people refer to often as the great plate count anomaly. We know there's a lot more bugs in our gut than we can grow. And I think now that people realize that sequencing has limitations in the ability to really know what bugs are doing, people are really starting to go after these now, and so I think that's going to get better. And I'll mention at the end, some of our next generation probiotics are going to be related to mutualistic. They're going to be mutualistic organisms, and some of those cause opportunistic infections, and so we'll have to think about how we're going to handle things like that. So one of the things I'll mention is that most of the studies that you see out there, we've seen a lot of this data today, are 16S community data. Not only are they 16S, they're partial 16S, they're small snippets. It's useful for inferring evolutionary relationships. It's not that useful for inferring function at a species or strain level. If there's one gene in the world that we know what it does, it's definitely this one. So you can't identify, and this is something I think is important, you cannot identify a species based on 16S sequence alone, especially partial 16S. So those of you who are taking your snippets and doing a blast and picking the first strain that comes up, don't do that anymore, because you're playing a pretty dangerous game, especially if you're using that as a basis for doing your future studies. It's a lot more complicated. It's very difficult to get past the genus level, depending on which strain you're using there. But people do this. You see this all the time in papers. Now, metagenomics will help, but I'll argue that it's really doing functional studies that is really going to be what drives therapies. We can go in and we can sequence the metagenome, but it's really a problem because even though we think we know a lot about functions of genes, I would argue that we don't know the biological function of at least half the genes that are in the microbiome, maybe more. Even the best studied strain, E. coli, the bioinformatics people will say, well, we have that 85 to 90% annotated, but the fact of the matter is that if you look at biological functions, it's probably half. And so until we start figuring out ways to get at this unknown gene content, the functions, it's going to be very difficult for us to really translate a lot of this. So just to prove the point that strains matter, species don't matter, let's say you have a bloom of E. coli in your microbiota. Is that going to be good or bad? So this is kind of let's make a deal. You can pick door number 1, door number 2, or door number 3. So, okay, who's picking number 1? Okay, not many people, almost nobody picks number 1 because they think I'm trying to trap them. But if you'd have picked number 1, you'd just have a garden variety commensal and you'd be okay. So now you guys are now down to 50-50 and you probably know what's coming. So who's got door number 2? Okay, well, sorry for you guys because you have E. coli 10157 and you're now on the kidney transplant list. And of course the third one is E. coli nissula. The point is that if you look at their 16S sequence, they're 100% identical. You don't know anything about that. And so just to show that it's not just E. coli, this is work from Jim Versalvik's lab at Baylor College of Medicine that we've done with him. This is just looking at relative levels of TNF stimulation from macrophages. He has a strain that we've been studying a lot that is anti-inflammatory. Here's another strain, again, 100% sequence identity, is in fact at the 16S level, has no effect on this inflammatory response. And in fact, we know that this is due to the acquisition of 3 genes that basically turn on histamine. Again, you would never know this from looking at your 16S sequence. So again, strains matter. And so if you're going out to find, you have a species and you want to go to the type strain, that might be okay. That might be a good place to start, but you probably need to isolate a lot of strains to hopefully get what you want. So also the other thing I think that's going to come up a lot is that a lot of the bugs that are really important members of our intestinal microbiota also are recognized by physicians as opportunistic pathogens, especially Bacteroides fragilis for anaerobic infections. But the fact of the matter is most of the times the only reason they're bad is because they get to parts of the body that they're not supposed to get to, not necessarily that that's what they're doing. So I would say that if you actually exclude everything that's in the gut like that from probiotics, I think we're going to be missing some key members and maybe some key opportunities. And you say, well, that's the way it goes. These things can cause disease. They can cause pathogens. But I would also say that that's what we're putting in when we do fecal transplants. So we're already doing this now. So if you're really that worried about it, then I think that we should be worried about it for fecal transplantation too. So these are just things that we think about. I'm not making any recommendations one way or another about how we're going to do it, but these are the kinds of issues that NIH, NCI, FDA are really going to have to think about when they start trying to regulate some of these organisms. And the fact of the matter is that we get it wrong early on, it's going to be even that much harder going forward. So just really to sum up, I mean, in the example I gave today is recurrent C. diff, but it really could be any disease here that we're now treating with FMT. And I will say FMT is not being just used for C. diff, it's being used for IBD. There are trials going on for obesity, type 2 diabetes. And some of these aren't so scientifically, I guess, valid to do. But, you know, these are going to make people happier. They're going to make people better. But the fact of the matter is if you think you can transmit health, we know from a lot of animal models that we can transmit disease, and some of these are listed here that have been shown in animal models. Now, you might argue that a 70-year-old patient who has recurrent C. diff, you may be not so worried about obesity or type 2 diabetes at that age, especially when they have no quality of life. But, you know, there's a lot of pediatric trials now for fecal transplantation for IBD and C. diff. That's a different story. These kids are now going to be raised for a lifetime with what you give them, and if we give them the wrong things, we may be setting them up for things that they might not normally be set up for. So I think that these are really important questions to answer. They're not easy to answer, but we're going to have to figure out ways to approach them so that, you know, we can get more to this side and minimize the risk on this end. So with that, I will take questions. This is my laboratory at Baylor. As I pointed out to people who did this already, we certainly thank funding from all the agencies, and I'll be happy to take questions.
Thank you. Great talk. So while doing the fecal microbiome transfer, some people get it, and those bugs are able to colonize it. With other people, those bugs are able to colonize, you know. So what is the role of host gut niche in that? I mean, I hardly see people talking about it.
The role of the host gut.
So what's the role of the specific, so the niche area, like the gut pH or host genetics, or, you know, what's the role of host in it? Well,
so what I will say about FMT, so there's more and more studies coming out now that some patients, they look like the donor for a period of time afterwards, and that sort of drifts away. Some donors, when you give them to patients, the patients within a few days don't look like them at all. Why? I don't think anybody really knows. And so there's a colleague at Baylor that likes to call, he thinks that really all these things do is just shake up the system so much. He calls it enslapment. I don't know where that came from, but he thinks that they really, that's what's really, it's just a shake up of the system. And one of the things that you can think of for FMT is that, as we've heard, there's so much individual variation in our microbiome, yet 90 to 95% of FMTs work. So there's some simple thing I think that's common that's going to be able to suppress, and whether it's enslapment or a single bug or a community of bugs, I don't think that's what people know. So that's really where I think things are going to go with FMT, but it's, you know, I think in the long run, if we can come up with a more defined community, in my opinion, that would be better to use. But I will say there are other people who think, no, that's a natural community. Anything you build in the test tube is going to be different. I don't know.
Very nice data. I have a simple question. I came away from some of the morning talks with what I already knew, that everything's my mother's fault.
I don't know your mom, so I don't get
You'd agree. But nonetheless, if you take two humanized mice, put them in a cage together in a micro-isolator, and have them have little pups, are those pups humanized, or do they regress back towards a mouse microbiome?
Yeah, they're humanized, and so that was one of the things we were interested in seeing over time. And so what happens is the initial animals that get the fecal gavage, so they were adult germ-free, they have a community. Now, they have their first offspring. That community kind of becomes a bit different, but then from then on, it basically stays the same, and it stayed the same for a few years. And initially, they were in isolators down at the University of Michigan, and then the person who was on the project didn't want to house the mice anymore, so we thought, well, gee, if we ship, I was at Michigan State at the time, so I said, if we ship them up to Michigan State, we're going to get them contaminated. And I mean, none of that happened, so I think that once you get a stable community in there, just like any stable community in any environment, it's just very hard to invade that.
And a quick technical question. In your bioreactors, is the media you feed it with, does that contain digestive enzymes and bile and?
Yeah, yes, sometimes. So we put in, we've used both bovine bile, we've used taurocholate. The digestive enzymes, I don't think we have digestive enzymes, but there are some people that put those in. It has it has some complex sugars, you know, but we don't have, we had, you know, any of the other simple sugars. So it's kind of I should just say it's actually built by Glenn Gibson and George McFarland. So the big 3-vessel reactor, we basically use the same media with just some tweaks. So,
excellent talk. Thank you very much. My question is also about the humanized mice. So what criteria did you use or to select whose or what bugs should represent?
Whose fecal sample should go in?
Yeah, like what should represent the human. And also, do the metabolic functions of that composition mimic that of human metabolic function? Like
So in terms of how we chose the fecal samples, so initially when we started all this work, we pooled samples. So we had 12 healthy donors. We pulled them together with the idea that we were worried if we just chose one, it might be a wacky one and we wouldn't know anything about it. Tried to publish the first paper, and all 3 of our reviewers said you should never pull feces, which I still don't understand why. So we went back and we've used individuals in both the reactors and the humanized mice. And in fact, they basically work either way. In terms of the metabolic functions, you know, I guess I don't really know how to assess that, you know, the entirety. I mean, you could sequence it and get an idea of what went in. What I can tell you is that in bioreactors and in mice, you know, we have a good amount of diversity that comes out, but we have some bugs that are very high in feces that become low in the reactors, and we have things that are low in the feces that become high in the reactors, so there is dynamics. A strength of the model, which I had, you know, again, reviewers didn't like much, there's actually variation which comes up, what kind of community comes up. It's not exactly identical over every reactor or every mouse, but what is identical is its ability to resist. And so, you know, I was worried that we just get one sort of defined community that might not be representative of anything. So it's not like it's totally random, but there is some, you know, stochastic variation in how, you know, what microbes get started, which is probably what happens in us. You just ate, depending on if you ate a cheeseburger or a salad, just now you probably are stimulating different bugs that are going to come up, and that might be different from person to person, depending on what you start with. So.
Okay, final question. Yeah, I think related to the same question, I think the question is how much is the difference between mouse microbiome and human microbiome, and when you pool the data from 12 donors, how different is the microbiome of each person? You combine those and then transplant into a mouse. So can you talk about the differences, original to begin with, what's the difference between the two, and then change?
Yeah, so I mean, for humans, it depends on who you have. And so for our group of people that we had probably a bit higher firmicutes than some other studies. For the humans, mice tend to be more dominated with Bacteroides. So when we actually put the human feces in there, we actually see that they're actually more Bacteroides that we had in our fecal samples grow up. So that, again, that kind of shifts. We do see that our mice don't respond exactly the same as traditional mice to the C. diff model, so we know that there are some differences. What I could also say about the mice is, again, that it's stable over time, so that doesn't really shift. And that was the second part of your question was the
difference between the two.
The individual. So, so I don't know that we've really looked at the data in terms of like using the pool versus the individual mice, because we just started these experiments again at Baylor. We've now colonized germ-free C57s, Swiss Websters, and Black 6 or Bald Cs. And we've done that now with not only just fecal, like normal, what we call normal microbiota, but also people who are on antibiotics. So we're going to start getting at some of those questions, hopefully, in the near future. So.
Okay, thank you, Rob.
Thanks.
And so the person whose phone went off when Rob was talking owes him lunch, so you can reimburse his lunch.
Really?
So, yeah.
Did somebody no, because they can take me to dinner, and I like expensive wine.
So.